So hello guys, welcome in another video tutorial of major histocompatibility complex playlist. So in this video tutorial we will going to discuss about the MHC class 2 molecule. We have discussed about the MHC class 1 molecule earlier. So if you have not watched yet, I would like to recommend you to go and watch this video. So now let's start the concept of MHC class 2 molecule. So first thing is, from where we will get the MHC class 2 molecule? We will get from MHC class 2 gene, right? So MHC class 2 gene giving us MHC class 2 molecule, that is the first thing. And as they play a role in antigen presentation, so the classification of classical MHC class 2, classical and non-classical MHC genes are applicable here, right? So we can divide MHC class 2 gene into two parts: classical MHC class 2 gene, non-classical class 2 MHC gene. Okay, now this classical MHC class 2 gene will going to take part in antigen presentation, non-classical will not going to take part in antigen presentation, right, as we have discussed in case of our MHC class 1. So now there are three important loci present. Okay, now the fact is, what is the meaning of loci? So for now you can just remember that loci is the meaning of the specific location of the chromosome, okay, where genes are present. Okay, that means the genes or class 2 MHC genes are present on three specific position of the chromosome, and these three specific positions are HLA-DP, HLA-DQ and HLA-DR. Okay, remember in case of class 1 MHC molecule we have talked about HLA-A, HLA-B and HLA-C, right? I hope you can remember it. So now HLA-A, HLA-B and HLA-C are genes. Remember it very carefully. But HLA-DP, HLA-DQ, HLA-DR that we are discussing here are loci. Okay, in those loci genes are present. Okay, so that is the idea. Please remember it. So in each loci there are two genes present, A and B genes. Okay, encoding alpha and beta chain respectively. Okay, so it is obvious that as we have seen in case of MHC class 1 molecule there are also alpha and beta chains, so it is quite expected that in case of MHC class 2 molecule we will again going to get alpha and beta chain. So they are coming from A and B genes respectively. Okay, for HLA-DP, HLA-DQ, HLA-DR, in each locus you will get A and B gene. Okay, now here there is a twist that in each locus there will be two kinds of gene. One is expressed gene, another one is unexpressed pseudogene. Okay, so one expressed gene for alpha and beta chain and unexpressed pseudogene. Now the question is, what is the meaning of pseudogene? So pseudogene, remember it very simpler words, that pseudogenes are genes that was once activated, or once they used to act as a normal gene, but during the course of evolution they become non-functional due to some mutation. Okay, so now remember pseudogene can be formed due to various type of reasons. Okay, there may be chance of duplication, there may be chance of formation of processed pseudogene. Okay, and there may be also due to lack of function of the proteins that those particular genes are giving. Okay, we will going to talk about pseudogene in some another playlist, but now for now you can just remember that in case of MHC class 2 molecule the concept of pseudogene is due to duplication. Okay, because remember in MHC class 1 molecules video I have told you that MHC gene has one another feature, that is MHC genes, many of the MHC genes are duplicated genes, right? So as a result, now try to think about this. You have both, let me think it how I can express I can present this to you. So now let's try to think about this, that you have one toy. Okay, so do you need same toy another? No, right? Similarly, in case of genes, it is applicable. Suppose the gene which are giving the protein that is responsible for particular type of protein production, okay, now that gene somehow gets duplicated, okay, and as a result what will happen, that particular protein will be produced in doubled amount. But do our body needs that excess amount of protein? No. So as a result, the duplicated gene will be inactivated. Okay. You may can also remember this in such a way. Suppose you are working in a industry for near about 15 years. Now a new guy with similar qualification and similar skills came into the office and apply for job. Then what your boss will tell him, that there is no post left for you, so sorry for that, because there are already one employee present here. So why a new employee for exactly the same function, right? So that is the concept of pseudogene. I hope now it is very clear to you. Okay, because here one express gene already giving us alpha and beta chain, so why one extra gene? That is why that extra gene becomes mutated and non-functional. Clear? So now in case of HLA-DQ locus, another important point, in HLA-DP there are two genes, DPA1 and DPB1, that together forms the DP molecule. Clear? So HLA-DQ are similar to HLA-DP. So there are also two gene present, DQA1, DQB1, that together giving us the DQ molecule. And here also the concept of expressed and unexpressed genes present. Okay, but in case of HRA-DR locus, there are 10 gene present. Among them, one is DRA gene and another one is 9 DRB genes present. Okay, one DRA gene and 9 DRB genes present. Clear? So now, although those things are not too much important, but this line is very important, that MHC class 2 molecule will going to present the antigen to helper T cell or T cell having CD4 marker, right? Because remember, in case of MHC class 1 molecule, I have told you that it will going to present the antigen to CD8 T cell. Okay, that means T cell having CD8 marker, and you can simply remember it with the help of that mnemonic, that MHC class 1 molecule, so 1 will going to interact with CD8, as a result you will get 8. Okay, so for 2 molecule, that is MHC class 2 molecule, it have to interact with CD4 marker containing T cells, so that it become 8, right? So that's how you can remember it, that in case of MHC class 2 molecule, it will going to interact with the T cell having CD4 as a surface marker, but in case of MHC class 1 molecule, it will going to interact with T cell having CD8 marker. Clear? So this is the structure of the MHC class 2 molecule. It is a little bit different from our MHC class 1, because remember, in class 1 molecule we have seen there was alpha 1, alpha 2, alpha 3 and 1 beta 2 microglobulin was present that interacts with alpha 3 via non-covalent interaction. That was the concept of MHC class 1 molecule, right? But in that case, there will be alpha 1 and alpha 2 domain in one chain and beta 1 and beta 2 domain in another chain. Okay, and remember, in case of alpha 1, in case of MHC class 2 molecule, the alpha peptides are 35 kilodalton and beta chains are 28 kilodalton. Clear? So these are the transmembrane segment. Cytoplasmic anchor segments are also present. Okay, so now, and another important thing, that in case of MHC class 2 molecule, the peptide binding groups are open at both end. Okay, but if you have watched the MHC class 1 molecule video, then I hope you can remember that in case of MHC class 1 molecule, these two domain was closed, right? That means the peptide binding group was open at one end. Okay, that is very important thing that you can mention when in your examination they will ask you to write about MHC class 1 and class 2 molecule difference. You can mention this point. Okay, so now this was all for that picture. Now let's move to the next slide. So two external domain, alpha 1 and alpha 2 domain in one chain, I have told you, beta 1 and beta 2 in another, and the membrane distal part here is alpha 1 and beta 1 that together interact to form the peptide binding group. Okay, so here these two are membrane distal domain, they are membrane proximal domain. So membrane distal domain alpha 1 beta 1 interact to form the peptide binding group. Clear? So now, so here glycoprotein CD4 will going to interact with the invariant portion of the class 2 MHC molecule, and that is obvious because in case of class 1 molecule we have seen that CD8 was interacting with the alpha 3 domain, which was the invariant part of the class 1 MHC molecule. So here CD4 will going to interact. Clear? And class 2 MHC molecule is a part of immunoglobulin superfamily. Okay, now in case of class 1 molecule, MHC molecule, I have told you that class 1 MHC molecule will be expressed on nucleated cell, right? So now antigen presenting cell are the cell that will going to express MHC class 2 molecule. Okay, so it is very important to discuss about the classification of antigen presenting cell. So remember, antigen presenting cell can be divided into professional antigen presenting cell and non-professional antigen presenting cell. So let's look at that slide, that is more clear. So now remember, professional antigen presenting cell, under professional antigen presenting cell you will get three type of cell: dendritic cell, macrophages and B cell. Okay, among them remember dendritic cell is the most efficient one. Okay, and obviously the powerful antigen presenting cell, and it will express MHC class 2 molecule at a high level constitutively. Okay, on the other hand, macrophage cannot express MHC class 2 molecule constitutively. First it have to be activated, then it can express class 2 MHC molecule. But in case of B cell, it can constitutively express class 2 MHC molecule but at a low level. Okay, that is the how they express MHC class 2 molecule. Okay, and all the professional antigen presenting cell differ by their location and migratory behavior. You may find some professional antigen presenting cells circulating throughout the body, and you may also find some antigen presenting cell residing in a specific tissue for a long period of time. Okay, but that is not important information. Here important information is dendritic cell is the most efficient and powerful antigen presenting cell, and it will going to express the MHC class 2 molecule constitutively at a high level. And on the other hand, macrophage have to be activated first, then it can express MHC class 2 molecule. And the last but not the least, that is B cell. B cell will going to express MHC class 2 molecule constitutively but at a low level. Okay, and B cell also having antigen specific surface receptor. It is another important point you can remember. And non-professional antigen presenting cell includes fibroblast, thymic epithelial cell, glial cell, pancreatic beta cell. They can be induced to express class 2 MHC molecule. Okay, now one thing I hope you have noticed, that in case of MHC class 2 molecule, you have to induce most of the cell to express class 2 MHC molecule, right? But in case of class 1 MHC molecule, it was not, right? Because in case of class 1 MHC molecule, we have already seen that all nucleated cell express class 1 MHC molecule, right? So that is the thing, please remember it, okay? But the exception is B cell. B cell express MHC class 1 molecule and class 2 molecule both constitutively, okay? That is a very important MCQ they may ask you, okay? Now another important concept, that the cell who are expressing MHC class 1 molecule will be known as target cell. Remember it very carefully, it will be very important, because after that video I will going to upload a video on how the antigens are processed before it is expressed by MHC class 1 molecule. That will be our next video's topic. So there I will going to use the term target cell. So before that it is necessary to understand. So all the cell that are expressing MHC class 1 molecule will be known as target cell, okay? On the other hand, all the cell that are expressing MHC class 2 molecule, most of the cases we are referring them as antigen presenting cell, okay? Clear? So now last topic of today's class, that is MHC class 2 peptide interaction, okay? So I hope you may remember that in case of MHC class 1 molecule, the peptide that was presented by MHC class 1 molecule to CD8 T cell was 8 to 10 amino acid containing, right? And in case of MHC class 2 molecule, remember the peptide will consist of 13 to 18 amino acid, okay? It will be presented to CD4 T cell, okay? Now the most important concept, that is in case of MHC class 2 molecule, remember the peptide will bind in this manner, okay? That means they are maintaining a constant roughly elevation on the floor of the binding groove. But in case of MHC class 1 molecule, if you remember, that was not, because there are specific amino acid sequence present on the peptide that was buried within the binding groove, okay? And the middle part of the peptide was arc away, right? So that was the confirmation how peptide will exist on MHC class 1 molecule, and this is the confirmation how peptide will exist in case of MHC class 2 molecule. That is very important to remember, and you can also use that point as a difference between MHC class 1 and class 2 molecule, okay? So that is the end of this video. So I hope this video will be helpful for you. We will again going to meet with a new topic of major histocompatibility complex, and I hope you are enjoying this playlist and it is becoming very helpful for you. So thank you for listening to this class. We will again going to meet in some another video. So till then, bye bye.